Uh, Hello everyone, I am Nafiseh Nasri. I am working in the therapeutic serum laboratory at the Razi Institute. This study was conducted in collaboration with Dr. Hossein Our study is about first investigation into Iranian Echis carinatus snake venom toxic fractions and their hematological impact. Echis carinatus inflammation, critical medical emergency. Snake venoms contain multifunctional protein complexes with diverse enzymatic activities targeting hemostatic and thrombotic system. This component exhibits synergistic or antagonistic effects on blood coagulation, rapidly incapacitating victims through complex hematological disruption. This mixture has component with synergistic or antagonist effects on blood coagulation. Apart from developing therapeutic agents from venomous animals can be studied for the presence of endogenous T-venom that protect against their own toxins without antivenom toxins. Without antivenom treatment, the disorder quickly worsen and this can occur soon. Here, we describe blood complexion caused by Iranian Echis carinatus venom and its fraction. Enzymatic component, multiple protein fractions with distinct coagulant and anticoagulant property targeting specific blood factors. Hemostatic disruption, venom components interfere with platelet aggregation, fibrinogen levels, and clotting cascade activation. Multiple target toxin acts stimulously on various protein target in prey, creating synergistic pathophysiological effect. In vitro and in-vivo coagulant assays were conducted to determine the effect of Iranian Echis carinatus venom and its fraction. In first step, venom fractionation, Iranian Echis carinatus venom separated into distinct toxic fractions using the advanced chromatographic techniques for individual analysis. In the second step, we also evaluate the in vivo and in vitro effects of the hematological analysis was used for determining the anticoagulant and procoagulant fractions. In the third step, we in vitro and in vivo effects of the polyvalent antivenom in neutralizing hematological manufactions. The HPLC method was used to examine the neutralization potency of antivenom. The result of the PT and PRPTT, TCT, FCT, hemorrhage, defibrinogenating platelet aggregation test, and hematological parameters for plasma reveal the multifunctional features of toxin which can act on the multiple protein targets of prey. In conclusion, the introduction of homotoxins fractions of Iranian Echis carinatus in this region could be of the great help in the production of an effective antivenom and also help the medical care department to find out the clinical signs of Iranian Echis carinatus venom. This report confirmed the Echis carinatus inflammation can create severe hematological disorder that become visible within the first hour after inflammation, which require medical and clinical attention as soon as possible to reduce the severity of clinical symptoms. In this slide, we show the HPLC result of venom, antivenom, and the mixture of venom and antivenom in different concentration of venom and antivenom. This figure shows the venom, the HPLC chromatogram of venom in 1 milligram per milliliter concentration. Antivenom, this figure is show the HPLC chromatogram antivenom. This figure show the mixture of venom and antivenom in this concentration. This slide there show the HPLC chromatography of a mixture of venom and antivenom in different concentration of venom. The concentration of the antivenom is stable. To the best of our knowledge, this was the first study to report the inflammation of toxic fractions of Iranian Echis carinatus venom in Iran. According to this study, some purified fractions are more toxic than the crude venom. Eventually, it was shown that the use of polyvalent antivenom leads to neutralization of enzymes and pharmacological properties of Iranian Echis carinatus venom. Conclusion and feature this direction, clinical impact, antivenom optimization, regional health care enhancement, foundational research for Iran. This landmark study establishing the foundation for improved clinical management of Echis carinatus inflammation in Iran. The identification of hemotoxic fraction and their neutralization by polyvalent antivenom provides a critical site for both antivenom development and emergency medical protocol. Thank you for your attention.